Good morning. Welcome to the first chapter, which is the history of microbiology. We're going to talk about the evolution of this discipline and the main protagonists that were involved in its development. First, we're going to start with why microbiology. And the short answer is money. The development of microbiology began in the mid to late 1800s. And at this time, vineyards were losing a lot of revenue to the spoilage of grape juice. They didn't know what caused fermentation. Effectively, what they would do is they would leave large vats of grape juice exposed to the air, and if natural yeasts landed in the grape juice, then they would end up with wine. If a bacteria landed on the grape juice, then they would end up with acidic fermentation, which was effectively spoiled. And if fungi landed on there, then there would definitely be spoilage. So a lot of money lost to allowing nature to bring about the correct type of fermentation. So what do? We cue in Louis Pasteur and his application of the scientific method in a biological discipline, okay? So here's Louis Pasteur. He was born in 1822, passed in 1895, and a world-renowned scientist. That was, again, the first one to apply the concept of the scientific method to microbiology. So, quick review. Scientific method. Observation, question, hypothesis, okay? So you have an observation. The observation usually causes us to ask a question about what we're seeing. And then that question generates educated guesses, which are called hypotheses. Once we have a hypothesis, a testable hypothesis in place, now we can go forward and derive an experiment that either supports or does not support the hypothesis. If this hypothesis is supported, fantastic. If it's not, then we modify the hypothesis until we come to the right conclusion. So that's the scientific method. And again, at that time, it was Louis Pasteur that applied it to the various hypotheses of the time. So what are the hypotheses? Well, one of them was that grape juice fermented spontaneously. In other words, on its own. Another hypothesis was that air caused it to ferment. Another hypothesis was that an organism caused it to ferment, but we didn't know what the organism was. So we had to test all these distinct hypotheses. And that's exactly what Louis Pasteur did, okay? If you look on this slide, you can see that he pasteurized the grape juice. In other words, he heated it enough so that there would be a minimal number of organisms in it. And then he stoppered it to see if it spontaneously fermented. In other words, to see if it fermented by itself. That did not happen. So that hypothesis was rejected. Then he used his swan-necked flasks to allow the grape juice to be open to the atmosphere to see if air was responsible for fermentation. That also did not take place. That hypothesis was rejected. Then he tried bacteria. That's what allowed him to realize that bacteria carried out lactic acid fermentation, and that is considered spoilage. That is not a desirable outcome, okay? And then the last one, of course, is pitching yeast into the grape juice. Now that carried out alcoholic fermentation, and that was actually what was desired. So due to Louis Pasteur's application of the scientific method to this question, we were able to derive that yeast is the actual organism necessary for alcoholic fermentation. As a consequence, Louis Pasteur is credited with being the father of the so-called industrial microbiology. microbiology. What is industrial microbiology? It is the use of microbes, bacteria, fungi, etc., in order to produce materials that you and I consume every day, such as cheeses, beer, wine, soy sauce, vinegar, yogurt, sour cream, artificial sweetener, bread. The list really goes on. Okay, so this is important. The fact that Louis Pasteur initiated this discipline of industrial microbiology. And I'm going to pause here for just a second, but I will return in just a second. Okay, we continue with a German scientist by the name of Buechner. And effectively what happened is that people were not satisfied that yeast was what was necessary for alcoholic fermentation. They wanted something even easier, something simpler. So Buechner came up with the idea of breaking yeast apart and see what happened. And the surprise was that if all the components of yeast were still there, then alcoholic fermentation would continue to ensue. Okay. What happened? Well, he discovered the fact that there's these things called enzymes. To you and me, these are called biological catalysts. And it is the study of these enzymes that started the discipline of biochemistry. And that's close to my heart because I'm a biochemist myself. So again, Buechner, a German scientist, was the one responsible for discovering enzymes. Enzymes, again, are biological catalysts. What's a catalyst? It is a substance that speeds up a chemical reaction without itself being changed or modified. Okay, and as a consequence, Buechner started the discipline of biochemistry. Very important. Next person that we're going to talk about is the incredibly accomplished Dr. Robert Koch. And Dr. Robert Koch started the discipline of etiology. In other words, what causes disease? Dr. Koch was actually a poor forgive me. As I was saying, Dr. Koch was a humble country doctor in Germany. And at the time, we had another problem taking place. And that problem was anthrax. So just like we had this issue with fermentation, where people were losing money because of spoilage, now we have this disease, anthrax, that's killing the beef industry, is killing cows. So it was very important to find out what caused anthrax. And despite the fact that Louis Pasteur had become a world-renowned scientist, Robert Koch, a humble physician, chose to actually pursue this investigation. Very interesting. And interestingly, Robert Koch is the one that found the causative agent of anthrax. And I'm going to tell you, half of it has to do with the fact that he was a very intelligent man, and the other half had to do with the fact that he was very lucky. Okay, the reason that I attribute him with intelligence is because, opposed to what Louis Pasteur was using, which is liquid media, Robert Koch changed to solid media to grow his microorganisms. And even though this sounds trivial, that is actually what allowed him to be able to discern the causative agent of anthrax. What else? Well, once he figured out what caused anthrax, he became very well known in Germany. As a consequence, he moved from the country to Berlin the capital of the country, and the German government threw new lab assistants and money at him. So this man became started as a humble doctor and became a world-class scientist practically overnight by discovering anthrax. Now, I told you why he was smart. Why was he lucky? Because as it happens, anthrax is caused by one of the largest bacteria out there. And the reason that matters is because the microscopes at the time were not very good. So he was very fortuitous in having chosen a well, no, it wasn't a choice. That was the investigation of the time. But he was still lucky in investigating something that was visible with the optics of the time. Another thing that's important is the fact that anthrax was the first disease, the first causative agent that we discovered that caused disease. So this is where it began, was with anthrax. Another thing that's important for you to know is the fact that despite the fact that Robert Koch was involved in a lot of different studies of fermentation, the study of fermentation and spontaneous generation with Louis Pasteur was not one of them. Okay, so there's what Dr. Koch looks like. Okay, you have an assignment that involves him that is fantastic. And a Nobel laureate, very smart young man at the time. And look what he did, okay? Staining techniques, he took pictures of bacteria. He estimated the number of bacteria in a broth. He used autoclaving, he used petri dishes. In effect, this is contemporary microbiology. He's the one that started it, and we're using the same tools today. Very, very neat stuff. He also developed what's called the Koch's postulates, that we're going to go into in a second here. And it's important for you to know that what made him successful in discerning the postulates was his ability to aseptically transfer and isolate bacteria in solid media, okay? So if you haven't caught on by now, if it's in red, it's probably particularly important, okay? So again, it is his ability to aseptically transfer and isolate bacteria in solid media that led him to discern the postulates. So this is why solid media is so important. If the color, if the margin, if the elevation, if the shape of a colony is different, that tells you that you have a different microorganism. This is only discernible on solid media, not on liquid media. So you can see that this blood agar petri dish has, I don't know, 20 or 30 different microorganisms on it. Very interesting. Here's Koch's postulates, okay? This is what allowed us to cause the foundation of etiology, the causation of disease, okay? There's only 4 of them, and they're very logical. The first one is that the suspected agent has to be found where the disease is found, and it has to be absent in healthy individuals. That makes sense. Now what? Now that suspected causative agent needs to be isolated and grown outside the host. Remember, I told you what made these postulates successful was his ability to aseptically isolate these microorganisms in solid media. Without his ability to do that, the second step in the postulate would not have been possible. Once we have isolated that microorganism, okay, now something strange happens. We're going to put it back into a healthy, susceptible host to see if it causes a disease. Okay? And last, we should find that causative agent in unhealthy individuals, and we should not find it in healthy individuals. Like I told you, very logical, but this is the foundation of etiology. What happened after he published this in 1879? Well, the doors of etiology were open. Look at this. In 1879, Albert Neisser discovered gonorrhea, and then in 1880, the causation of malaria, typhoid fever, diphtheria, E. coli, and so the list goes on. So effectively, Dr. Koch opened the doors to etiology to all of these scientists because they all applied his four postulates to the study of causative agents. Very neat stuff, okay? And I'm gonna take a little. All right, let's introduce one more important protagonist here, and that would be Florence Nightingale. Again, this is one of your assignments, so hopefully you're going to learn a little bit about her. But Florence Nightingale, she was nobility, she was wealthy, she was somebody that really had the entire world open to her. And in 1854, the Crimean War between the British and the Ottoman Empire was going full board, and the British forces were losing a lot of men, but they were not losing them to bullets. They were losing them to cholera. They were losing them to fevers and effectively to infectious diseases. So your chances of being killed in the battlefield was somewhere around 20%. But your chances of dying in a hospital from an infection was somewhere around 60%. So your chances of dying in the hospital were much better than in the field. And they recognized that this was a problem. They had nurses before, but the women were untrained, and they had a tendency to cause more problems than they solved. So the Secretary of War called Florence Nightingale in. Florence came in, and I got to tell you, without formal education, because even when women went to college back then, they did not have formal education. Florence Nightingale went in there with a beautiful mind, and she saw the squalor and the dysentery and patients lying in their own stool and urine, and acknowledged that this was unacceptable. So with 34 nurses, she went in there, she grabbed the convalescing patients and her nurses, and with scrub brushes, she cleaned the entire hospital, brought in new sheets, separated men from their clothing and from their bandages. This is called the isolation of fomites. In other words, the isolation of infectious materials. And she introduced treatment of water. She introduced the better nutrition for the patients. And as a consequence of everything that she did, the death rate in the hospital was lowered by 2/3. No small accomplishment. But not only did she do this, she also journaled all of this. She accumulated data showing what she was doing and what the effect was. This is what made her particularly intelligent, because she could now demonstrate from an evidence basis that what she was doing was actually effective. So this is a very bright lady. When she came back to England, she got a hero's welcome. The Queen gave her about $250,000 back then, which was a ridiculous amount of money. And with that money, she actually founded the first nursing hospital. So her devotion to her calling never stopped. An incredible woman. Okay, so here we have it. We started with Pasteur, which is industrial microbiology, Buchner, which again started biochemistry, Robert Koch, which initiated the discipline of etiology or causative medicine. Let's see. Environmental microbiology came in later on with Bajorkian, Graham. Hopefully you've heard of the Graham stain. He started microbial morphology, and on the list goes, okay? So all these are contributors to the discipline of microbiology. Okay, a couple of people that we need to talk about is Kluver and Neal. So Kluver and Neal studied microorganisms, studied the metabolism in microorganisms, and made the argument that their studies could be applied to every other living system, including ourselves. And that's actually pretty good, because as it turns out, glycolysis, Krebs cycle, electron transport chain, all those main aspects of metabolism are actually very close to identical in both microbes and ourselves. So again, Kluver and Neal responsible for using microorganisms as model systems for biochemical reactions in all other animals. Okay. Just one more thing I want to address. You can read through this if you want to. It's pretty good material. But the last thing I wanted to talk about is bioremediation, and does the use of microorganisms, bacteria, fungi, algae, to break down pollutants in nature. So for example, if we have an oil spill, you can actually add bacteria, and the bacteria can break down the oil into simple organic molecules that do not destroy the organism, the ecosystem. So bioremediation is our use of these organisms in order to detoxify and maintain the environment in a safer condition. Okay, so that's it for this chapter. I hope you learned a little bit, and I'll see you soon again. Okay, thank you for your time.